Welcome to the deep dive. Today, we're tackling a really fundamental area in radiation oncology, DNA repair.
Right.
We'll be focusing specifically on base excision repair, BER, and nucleotide excision repair, NER, two, key pathways cells use to deal with DNA damage.
Exactly. Think of it as getting under the hood, understanding the microscopic mechanics that really influence how our treatments, like radiation, actually interact with cancer cells.
Right.
We'll explore how these pathways work and, crucially, what that means for us in the clinic.
And given that radiation works by, well, inducing DNA damage, understanding these repair mechanisms is absolutely vital for anyone in radiation oncology, wouldn't you say?
That's foundational stuff.
Okay, let's dive into base excision repair, or BER then. What's its main job?
So base excision repair, BER, is like the cell's daily spell checker for its DNA. It's mostly concerned with fixing small, kind of localized errors affecting single bases.
Single bases.
Yeah, imagine finding and correcting just one misspelled letter in a huge book. These errors can be damaged or incorrect bases caused by things like ionizing radiation, think thymine glycol or 8-oxyguanine, but also spontaneous chemical changes like deamination, which just happens.
So it's dealing with the more subtle types of DNA damage, the small stuff.
Precisely. Single-letter typos. And BER is generally thought of as a high-fidelity pathway.
Meaning,
It usually corrects these problems without introducing new errors. It's pretty accurate.
interesting. So how does the cell actually do this? How does it fix these single base errors? What are the steps in BER?
Okay, so the BER pathway, it's a really coordinated sequence of enzyme actions. Step one is recognizing and actually removing the damaged base.
Right.
This is done by a family of enzymes called DNA glycosylases. Think of these as specialized tools. Each one is designed to spot and grab a specific type of damaged base. There are quite a few different glycosylases.
Like a set of specialized tweezers, maybe, for different kinds of errors?
That's a great analogy, yeah. The glycosylase essentially flips the damaged base out of the DNA helix.
Flips it out.
Yeah, flips it out, and then it cuts the glycosidic bond that's the link between the base and the sugar phosphate backbone.
Okay.
And that leaves behind what's called an AP site. It stands for a pyridinic or a pyrimidinic site. Basically, a spot on the backbone where the base is just missing.
A gap.
A gap where the base was.
Yeah.
Now, some glycosylases also have what's called like ACE activity.
Base?
Yeah, meaning they can also make a little nick in the DNA backbone right there at the AP site, sort of a 2-for-1 deal.
Okay, so the damaged base is gone, leaving this AP site, this gap. What happens next?
Well, if the glycosylase didn't already make that cut, the next step is backbone incision. An enzyme called AP endonuclease 1, or APE1, comes along.
APE1?
Yep, APE1 recognizes that baseless site, the AP site. And APE1 then cuts the phosphodiester backbone, the structural part of the DNA, right next to this AP site, creating a single-strand break.
Okay, so a break is introduced. Now the cell needs to fill that gap with the correct base.
Exactly. And this brings us to the gap-filling stage. There are two main ways this can happen: short patch, BER, and long patch, BER.
Short patch and long patch.
That's right. Short patch BER is the more common route by far. Yeah. Here, an enzyme called DNA polymerase beta, pullback, comes into play. Now, pullback has this really neat ability. It can remove a little sugar phosphate bit left behind after APE1's cut and simultaneously insert the correct nucleotide into the gap.
Ah, so it cleans up and fills in one go.
Pretty much, yeah. And a really key player in this short patch process is a protein called XRCC1.
XRCC1, okay.
Think of XRCC1 as a coordinator, maybe a scaffold. It helps bring pullback and the final sealing enzyme together, make sure they work efficiently right there at the break.
And the sealing enzyme?
That would be DNA ligase 3 or LIG3. It seals up the nick, restoring the continuous DNA strand. Job done, usually.
Okay, that's short patch. You mentioned long patch BER, too. When does that happen?
So long patch BER is less frequent. It's typically used for certain types of damage, maybe some oxidative lesions, or maybe if short patch BER gets blocked somehow.
Right.
Instead of just replacing one nucleotide, it involves displacing a slightly longer stretch of DNA, maybe 2 to 10 bases.
Okay, a bigger patch.
Slightly bigger patch. And it uses different DNA polymerases, pol delta or epsilon, along with those replication helper proteins like pCNA and RFC.
The ones we see in DNA replication.
Exactly. Then an enzyme called flap endonuclease 1, FEN1, comes in and snips off that displaced flap of DNA containing the bad sequence. And finally, DNA ligase I seals the nick.
Ligase I, not ligase third like in short patch.
Correct. Different polymerase, different ligase for long patch.
Okay, so short patch is the quick fix, single base, pol beta, XRCC1, ligase 3. Long patch handles slightly trickier stuff. Needs pol delta epsilon, PCNA, FEN1, Ligas 3.
That's a good summary. The main players in BER overall. DNA glycosylases started off, then APE1, XRCC1 is crucial, mostly POLB, and mostly LIG3.
Got it. And you mentioned an overlap with another pathway.
Yeah, it's worth noting that BER does overlap somewhat with single-strand break repair, or SSBR. We'll probably talk more about SSBR later. But proteins like PRP, that's poly-ADP ribose, polymerase, and XRCC1, which we just mentioned for BER, are involved early in SSBR too. And another enzyme, polynucleotide kinase, or P&K, helps clean up damaged ends in SSBR, so there's some crosstalk there, especially in the later steps.
Fascinating how these systems interconnect. Okay, so that covers the smaller DNA errors, the single base problems. What about when the damage is bigger, more substantial, causing a real distortion in the DNA structure? That's where nucleotide excision repair comes in, NER.
Exactly. Nucleotide excision repair, NER, is the pathway that deals with the bulky stuff, lesions that significantly warp the DNA helix structure.
Okay.
If BER is like correcting a single typo, NER is maybe more like cutting out and replacing an entire damaged word or even a short phrase that's messing up the sentence structure.
That's a helpful analogy. So what kind of damage actually triggers NER?
Well, the classic example everyone learns is pyrimidine dimers.
Like TT dimers from UV.
Precisely. Thymine dimers caused by ultraviolet radiation from sunlight. But NER also handles other bulky chemical adducts, chemical groups latching onto the DNA.
From what?
These can come from certain chemotherapy drugs, like, say, alkylating agents or platinum drugs, or even from environmental carcinogens, like components in tobacco smoke.
Okay, bulky adducts. Now, this seems like a really key point for us in radiation oncology.
Yes.
Does ionizing radiation cause these bulky lesions that NER fixes?
Good question. And the answer is generally no. Ionizing radiation, the kind we use in therapy, does not typically cause the bulky helix-distorting lesions that NER is specialized for.
Right.
Ionizing radiation mainly leads to that single-base damage handled by BER, as we just discussed, and also single-strand breaks and, importantly, double-strand breaks, which other pathways handle.
That's a really important distinction to keep hammering home. BER for RT-type base damage, NER for UV and bulky adducts.
Exactly.
So how does NER find and fix these bigger distortions then? It sounds more complicated.
It is a bit more intricate, yeah. It involves around 30 different proteins, and it operates through two main sub-pathways, depending on how the initial damage is spotted.
Okay.
There's global genome NER, or GGNER, and transcription-coupled NER, or TCNER.
Global genome and transcription coupled, what's the difference?
So GGNER acts like a genome-wide surveillance system. It's constantly scanning the entire genome for any helix-distorting lesions anywhere.
How does it spot them?
The initial recognition, the damage sensor, is mainly this complex called the XPC complex. It includes the XPC protein, HR23B, and centrin-2. Sometimes another protein, XBE or DDB2, helps out.
XPC complex.
Right. This complex basically bumps along the DNA, feels for a distortion, binds to it, and flags it saying, hey, problem here.
Okay, that's the global scan. What about TCNER, transcription coupled?
TCNER is more targeted.
And faster.
It specifically deals with lesions that happen on the template strand of genes that are actively being transcribed.
The genes being used right now.
Exactly. Think of it as prioritizing the repair of the important instruction manuals the cell is currently reading.
Makes sense. How does that work?
Well, when RNA polymerase, the enzyme that reads the DNA to make RNA, is chugging along and hits one of these bulky lesions, it stalls. It gets stuck.
Okay.
Specific proteins then recognize this stalled polymerase. The key players here are CSA and CSP. These are the proteins defective in Cockayne syndrome A and B.
Ah, the Cockayne syndrome proteins.
Right. They recognize the traffic jam, the stalled RNA polymerase, and they help recruit the rest of the NER machinery right to that spot. Get those critical genes fixed quickly so transcription can resume.
That's clever. Prioritizing the active regions. So once the damage is found either by GGNER scanning everywhere or TCNER finding a stalled polymerase, what happens next? Are the steps similar then?
Yes, the core mechanism after damage recognition is pretty much the same for both subpathways. First up is DNA unwinding.
Unwind.
You need to open up the DNA around the lesion. A big multi-protein complex called TFIH is recruited. Interestingly, TFIH also plays a role in starting transcription.
Multitasking complex.
Very much so. TFIH has helicase activity, meaning it can unwind DNA. Specifically, its XBB and XBD subunits unwind the DNA around the lesion, creating a bubble of maybe 24 to 32 nucleotides.
Opens it right up.
Opens up. And other proteins like XPA and RPA, replication protein A, jump in to help stabilize this open structure and also double check that there really is damage there.
Verification. Okay, DNA is open, damage confirmed. What's the next step? Cutting it out.
Precisely. The next critical step is dual incision. Two different enzymes, endonucleases, make cuts on the damaged DNA strand, one on each side of the lesion.
All cuts.
Two cuts. XPG cuts on the 3-foot side downstream of the lesion, and the XPF-ERCC1 complex cuts on the 5-side upstream.
XPG and XPF-ERCC1.
Yep. These two cuts effectively excise or cut out the damaged oligonucleotide, that short stretch of DNA, usually 24 to 32 bases long, that contains the bulky lesion.
So the whole damaged segment gets snipped out.
Snipped out and removed. Then, of course, you have a gap.
And you need to fill it.
Exactly. Gap filling time. This is done by DNA polymerases, delta and epsilon again, the replicative polymerases, along with their buddies PCNA and RFC.
Just like in long patch BER, kinda.
Similar machinery, yes.
Yeah.
They use the undamaged complementary strand as a template to synthesize the correct DNA sequence, filling in that gap.
Okay.
And finally, DNA linkshotide comes in and seals the last remaining nick in the backbone, completes the repair.
Wow, that's quite a production line. A lot of players. You mentioned the XP proteins, XPC, XPG, XPF. What's the clinical significance there? That sounds important.
Yes, absolutely. You notice all the XP designations, XPA, XPC, XPE, XPE, XPF, XPG. That XP stands for xeroderma pigmentosum.
Ah, the disease.
Exactly. It's a rare genetic disorder. And defects in any of these XP genes lead to a faulty NER pathway.
And the result?
Patients with xeroderma pigmentosum have an extreme sensitivity to sunlight, particularly the UV component. They burn incredibly easily, and tragically, they develop skin cancers, multiple skin cancers, at a very high rate, and often at a very young age.
Because they just can't fix the UV damage.
Precisely. They can't repair those pyrimidine dimers we talked about. Similarly, defects in those other NER-related genes, CSA and CSB, cause Cockayne syndrome.
Which also involves UV sensitivity.
Yes, UV sensitivity is a feature, but Cockayne syndrome also comes with significant developmental delays and neurological problems. It's a multi-system disorder. But both these conditions really starkly illustrate just how critical NER is for protecting us from environmental DNA damage, especially UV, and preventing cancer.
These genetic syndromes really drive home the importance, don't they? Okay, let's pivot to the clinic now. For our listeners, the radiation oncology residents and practitioners, what are the clinical implications of defects in BER versus NER? Thinking about cancer risk, treatment response.
Right, that's the crucial link. Understanding these defects helps us understand why some people might be more prone to certain cancers, or how their tumors might respond differently to our therapies.
Let's start with BER defects then. What are the clinical implications there if BER isn't working right?
Okay, so BER, because it mainly handles ongoing damage from like normal metabolism, reactive oxygen species, and some environmental stuff, defects in BER generally don't cause dramatic sensitivity to radiation or most standard chemotherapy.
Not like NER defects in UV?
Not usually, no.
Okay.
However, BER is constantly working, fixing thousands of spontaneous base lesions every day in every cell. So if it's deficient, you can get an increased accumulation of mutations over time,
Leading to...
potentially contributing to cancer development down the line. It's got
Yeah.
a higher background mutation rate. Now, there is a notable exception we should mention, XRCC1.
the scaffolding protein.
Exactly. Remember, it helps in both BER and single-strand break repair. Well, mutations in XRCC1 have been linked to a slight increase in sensitivity to ionizing radiation. It's not dramatic, but it's there.
That's interesting. So XRCC1 is a bit different.
It is. And clinically, this whole area is relevant with PRP inhibitors.
Right, PRP inhibitors.
They're drugs used in cancer treatment, and they work by targeting DER and SSPR pathways. The idea, often called synthetic lethality, is that if a cancer cell already has a defect in another repair pathway, like homologous recombination, hitting B or SBR with a PARP inhibitor can be lethal to that cancer cell, while relatively sparing normal cells.
Clever. Exploiting an existing weakness. Okay, what about NER defects then? How do they manifest clinically, especially in our patients undergoing treatment?
Right, NER defects. This is where you see that profound sensitivity to UV radiation, as in xeroderma pigmentosum. Severe sunburns, tons of skin cancers, very young onset.
Okay.
And, importantly for chemotherapy, NER defects also lead to sensitivity to drugs that create bulky DNA adducts.
Like which ones?
Things like alkylating agents, cyclophosphamide is an example, and platinum-based drugs, like cisplatin and carboplatin.
Cisplatin, very common.
Very common. So if a tumor has defective NER, it might be more sensitive to those particular drugs. But, and this is the crucial point again for radiation oncology, NER defects do not typically cause a significant increase in sensitivity to ionizing radiation.
Why not? Just to reiterate.
Because, as we said, ionizing radiation mostly causes base damage, BER's job, single-strand breaks, SSBR's job, and double-strand breaks, handled by NHEJ and HR. It doesn't generally create the bulky, helix-distorting lesions that NER is built to fix. Different damage types, different repair pathways.
That distinction is absolutely key. And you also mentioned NER playing a role in fixing interstrand crosslinks. Can you expand on that?
Yes, briefly. Interstrand crosslinks, or ICLs, are nasty lesions where the two DNA strands get chemically stuck together.
Prevents strand separation.
Exactly. Blocks replication and transcription. Very toxic. NER does play a role in initiating the repair of ICLs, particularly if they occur in the G1 phase of the cell cycle. However, if an ICL forms during S phase, when DNA is replicating, it causes the replication fork to stall. This stall activates a different, more complex pathway involving the Fanconi anemia protein complex.
The Fanconi anemia pathway.
Right. This pathway coordinates multiple repair systems, including NER components and also homologous recombination, HR, to resolve the crosslink.
So it's a team effort involving NER and HR in S phase.
Pretty much. And that's why cells or patients with defects in either NER or HR pathways, like in Fanconi anemia itself, which involves defects in those coordinating proteins, show increased sensitivity to cross-linking chemotherapy agents.
Which agents specifically?
Things like mitomycin C and, again, the platinum compounds like cisplatin. Their effectiveness relies partly on overwhelming these cross-link repair pathways. It really highlights how interconnected everything is.
Absolutely. This is all incredibly relevant for understanding cancer biology and how our treatments work, or sometimes don't work. Okay, let's try and boil this down. What are the most important points for listeners to take away? Let's do our clinical pearls.
All right, clinical pearls. Let's nail these down. Clinical pearl number 1, base excision repair, BER. Think small lesions, damaged bases, incorrect bases from radiation, oxidation, spontaneous stuff. Key enzymes. Glycosylase is started, APE1 cuts the backbone, proline only fills the gap, XRCC1 is the scaffold, and LIG3 mostly seals it.
And the clinical consequence of defects.
Generally, increased mutation rates over time, potential cancer risk. But remember the exception. XRCC1 defects can cause slight radiosensitivity. That's important.
Okay. Pearl number 2.
Clinical pearl number 2, nucleotide excision repair, NER. Think bulky, helix-distorting lesions. UV dimers are the classic, but also chemical adducts from some chemos or environmental toxins.
Key players.
Key players. XBC complex for global scanning, GGNER, CFSESB for fixing transcribed genes, DCNER, the TFIH complex with XPBB, XPD for unwinding, XPG and XPF-ERCC1 for the cuts, pol delta or epsilon for synthesis, and LIG1 for sealing. Remember those XP proteins.
Got it. And the third pearl.
And clinical pearl number 3. NER defects mean extreme sensitivity to UV light. High skin cancer risk, think xeroderma pigmentosum, XP, Cockayne syndrome. Also sensitivity to bulky adduct-forming chemo like cisplatin. However, NER defects generally do not cause significant sensitivity to ionizing radiation.
Because RT causes different damage types.
Exactly. Keep that BCR versus NER damage type distinction clear. It matters for radiation oncology.
Excellent. Those are really crucial points. Now let's see if that's stuck. Ready for a quick board review blitz?
Let's do it. Fire away.
Question 1. A young patient presents with a history of severe sunburns after minimal sun exposure and has already developed multiple basal cell carcinomas, a defect in which DNA repair pathway is the most likely cause. A, BER, B, NHEJ, C, NER, D, HR.
Okay, severe UV sensitivity, early skin cancers. That screams NER. So C, nucleotide excision repair.
Correct. Question 2. Which enzyme kicks off base excision repair, BER, by recognizing a specific damaged base and cutting the bond between the base and the sugar, creating an AP site? A, APE1, B, pol-tup, C, LIG3, D, DNA glycosylase.
The initiator of BER, removing the base, that's the job of DNA glycosylase.
Correct again. Question 3. A cancer cell line shows unusual resistance to cisplatin, a drug known to cause bulky DNA adducts. Lab tests find a problem with unwinding the DNA around these adducts during repair. Which complex is most likely deficient? A, ORC, origin recognition complex, B, XPF-ERCC1 complex, C, TFI-IH complex, D, pol delta epsilon.
Unwinding DNA for NER, that's the helicase activity within the TFI-IH complex using XPB and XPD.
Right on. Final question. A patient has a known mutation in a DNA repair protein. They show slightly increased side effects from radiation therapy compared to average. Genetic testing points to a protein involved in both BER and SSBR scaffolding. Which protein is it? A, APE1, B, pol beta, C, Lig3, D, XRCC1.
Okay, slight radiosensitivity involved in both BER and SSBR scaffolding role that points strongly to DXRCC1.
Excellent. You nailed them. That really reinforces those key concepts we've been discussing. So just to wrap up, what's the big picture takeaway on BER versus NER for radiation oncology?
I think the main thing is BER is the primary pathway, dealing with the kind of single-base damage that ionizing radiation often causes, plus all the background metabolic damage. NER, while absolutely critical for handling UV damage and bulky chemical adducts, isn't the main player when it comes to repairing typical RT-induced damage.
Right.
Understanding that difference helps explain sensitivities, resistance. It's fundamental.
And knowing about the clinical syndromes linked to defects like XP or the slight radiosensitivity with XRCC1 gives us important context for our patients.
Precisely. These basic science mechanisms have real clinical relevance, impacting everything from cancer risk to treatment response. It provides that vital framework.
This has been incredibly informative, a really good deep dive into basic excision repair and nucleotide excision repair. Thanks so much for breaking it down.
My pleasure. It's foundational, like you said, and really essential knowledge for anyone in our field.
Absolutely. Join us next time on The Deep Dive, where we'll continue our journey through DNA repair, focusing on single-strand break repair and the critical pathways that handle double-strand breaks, NHEJ and HR, essential for understanding radiation's full impact.
Looking forward to it.
Until then, keep exploring the fascinating molecular world that underpins radiation oncology.